A silent threat is spreading across the globe, and it's something you've likely never heard of: latent antimicrobial resistance. Researchers have discovered a widespread, hidden reservoir of bacterial resistance to antibiotics, lurking in wastewater around the world. This is a critical finding, and here's why you should care. Scientists analyzed 1,240 wastewater samples from 351 cities across 111 countries. The study, spearheaded by the DTU National Food Institute in Denmark, revealed that these latent antimicrobial resistance genes are present on every continent. While they don't currently pose a major threat, experts believe some could evolve into serious problems in the future. But here's where it gets controversial. The research, published in the prestigious journal Nature Communications, suggests that we need to significantly broaden our surveillance efforts. The study's lead authors, Hannah Marie Martini and Associate Professor Patrick Monk, emphasize that we're dealing with a far more extensive problem than previously imagined. What exactly are these latent genes? They're essentially the blueprints for antibiotic resistance, but they aren't actively causing infections yet. The researchers compared the geographical distribution of these latent genes with the acquired resistance genes, the ones already causing problems, and found that the latent ones are far more widely dispersed. To curb future antimicrobial resistance, we believe that routine surveillance of antimicrobial resistance in wastewater, in addition to including already acquired resistance genes, should also encompass latent resistance genes, in order to account for tomorrow's problems as well, says Patrick Monk. Interestingly, the study confirmed that acquired resistance genes are more prevalent in Sub-Saharan Africa, South Asia, and the Middle East/North Africa, MENA, regions, a pattern consistent with previous research. The hope is to prevent a future pandemic. As the World Health Organization (WHO) has already declared antimicrobial resistance, AMR, a pandemic, the stakes are incredibly high. Acquired resistance genes are already making it difficult, and sometimes impossible, to treat infections with antibiotics. This new research offers a chance to get ahead of the problem. By monitoring both acquired and latent resistance genes, researchers can gain a comprehensive understanding of how these genes develop, spread, and jump between bacteria. Wastewater is a particularly useful monitoring tool because it contains waste from humans, animals, and their surroundings. Martini points out that there are more latent resistance genes globally than acquired ones. The team believes that some of these latent genes will eventually cause problems. The ability to predict which ones, and how they spread, could revolutionize our ability to combat antibiotic resistance. When new antibiotics are developed, a process that takes many years, bacteria may already have invented new scissors capable of destroying them. If we can study both types of genes over time, we may be able to find out which of the latent genes become problematic resistance genes, how they arise and how they spread across geography and bacteria, and in that way lessen the burden of antimicrobial resistance, says Patrick Monk. So, how do scientists identify these latent genes? The process, called functional metagenomics, involves extracting DNA from a sample and testing random fragments to see if they can confer antimicrobial resistance. It's like a scientific treasure hunt, looking for the genetic keys to unlock resistance. The key difference between latent and acquired resistance is that acquired genes can already jump between bacteria in the environment. Latent genes can do this in the lab, but their ability to do so in the real world is still unknown. Our concern is that some latent resistance genes will become acquired resistance genes, and thus become able to jump to different bacterial hosts out in the environment. Especially because the research also shows that they are present in large numbers in so many places around the world. That is why we would like to see them included in surveillance, says Patrick Monk. What are the environmental pressures that drive antimicrobial resistance? Antibiotics in the environment, from hospitals, agriculture, and wastewater, kill off susceptible bacteria, giving resistant ones a survival advantage. Disinfectants and biocides can also select for resistant bacteria. Here's a question for you. Do you think it's important to monitor latent resistance genes, even if they don't pose an immediate threat? Share your thoughts in the comments below.